wiggling around and stuff. In here, that hasn't happened yet. You'll find out what the story is in that in a few minutes. So this is the process of spermatogenesis.
So at the barrier, they're diploid, and then when they get
up here they're diploid, and as they move in this direction, so to speak, then they become haploid because this is where all the reductive division is taking place.
Okay.
There's another term on here that I also want to point out to you, and I'll show you. I won't try to show it to you on a slide because I really can't reproducibly find them. But inside the seminiferous tubules, there are special little cells, which I think have a very nice name. They're called nurse cells. Okay? They actually have a more formal name than that. But you know, the idea of a nurse cell and what they do, I think, is really kind of really cool when you learn what they do. For this process to take place, you have to have a chemical for spermatogenesis to occur. And that chemical is called testosterone. It has to be produced. In other words, if you have no testosterone, this process will not occur. All right. Now, one thing I haven't told you about yet, and you'll see in a slide, but you can see it on here, is bordering very closely near many of the seminiferous tubules, there are other specialized cells called interstitial cells. I'll show you a better picture of it in just a second. All right, interstitial cells do one thing. They produce testosterone. Right? They produce it. Where's it needed? Right here. Right? So it's produced here, just diffuses across this little membrane here, and allows spermatogenesis to occur. But even more remarkable than that is the job of the nurse cells. The nurse cells actually bind testosterone. In other words, the stuff which is made here and is required here for this process to occur is actually bound by the nurse cells so that the concentration is even higher. And so it's a really cool little kind of juxtaposition of, you know, where things are needed, where they're produced, and everything else. So the nurse cells bind testosterone, which is produced by the interstitial cells, which allows spermatogenesis to occur. So here's another picture. Got a couple more pictures. Here's one. Don't like this picture very much, but again, as Patrick was saying, the process out here with the stem cells, these are diploid, and as we move in this direction, they become haploid, and these would be mature sperm cells, 23 chromosomes, but they're not wiggling around yet. You're going to have to wait a little bit to do that. So here's a summary of this and then a couple of pictures. The term spermatogenesis is real simple, just means sperm production. It occurs in the seminiferous tubules in the testes. It's stimulated by testosterone, which is produced by interstitial cells in the seminiferous tubules themselves, in the testes. But the whole thing is ultimately controlled by a hormone produced by the pituitary gland, and the pituitary gland I'm speaking about now is actually the one in your head. Okay, it's called luteinizing hormone, LH. Right? Well, actually, LH is also required for the female reproductive cycle for oogenesis. So at the start of puberty, what really happens is luteinizing hormone is released, which then stimulates the beginning of testosterone production in the testes. Now, yeah?
For the mature sperm, how is the flagella formed? Where does it, I mean...
That's a really good question, and you know, one I'm not sure I have an answer to, but we can go back here. Just in the process of division, how to put it, the genetic material, the DNA which is found in there, actually instructs the cells, the sperm, to actually produce flagella. Yeah, it's not something which is made elsewhere and somehow attaches or something like that, which is not unusual to happen in biology. It's just part of the developmental process. Yeah, so it just happens in the testes.
Genetically designed.
Yeah, exactly. We've seen this one. Let me skip through this one. Here's a picture I want to show you. This will look similar to the picture I'll show you at the end of this little presentation. All these little round things are seminiferous tubules. And then you can see interspersed kind of like, you know, in between, kind of like this. Let me see if I can draw a picture. Here it is. So, you know, if this is a seminiferous tubule, and here's another one, and here's another one, and here's another one, and another one here. what you find irregularly spaced in around these circles, are little patches of cells, like maybe here and up here, which they're kind of pointing out like here and here and maybe some more there. Those are little patches of the cells called interstitial cells. And those are the cells which are producing testosterone, which just sort of diffuses all through here and allows spermatogenesis to occur. So that's one view of them. I'll show you a picture of what they look like. Here's another closer view. Here's just one part of a seminiferous tubule, and you can see where they, this is the name. I don't use this name, Leydig cells. This is the old name for them. They're called, Leydig cells are the same as interstitial cells. And so those are the cells which are producing testosterone. So that's pretty cool. We don't worry about Sertoli cells or any of that kind of stuff. So this is what you'll see on the slide that I'll show you in a little while. It's just another picture. These would be mature sperm cells in here. I really don't know what the red and blue arrows are pointing to. I really don't care. So anyway, it's the process of spermatogenesis. Oh, this next slide, you don't have to bother with this next slide. Somebody asked me this question the other day, so a couple of semesters ago, so I stuck it in. But it's not on the test. But somebody asked me about testicular cancer, and I'm going, I don't know much about it. I got a couple of slides in here tonight that people just asked me questions about, so I put them in. But it's the data I got for testicular cancer, maybe as many as 8,000 cases a year. You can see the age group that it's most common in. But I think the important thing to see here on this slide about testicular cancer, and especially if you're a nurse or if you ever know anybody that has it, it is one of the most curable cancers. A cure rate of 90%. Sometimes the testis, either part or all of it, may have to be removed, and then typically, radiation or chemotherapy. But the actual risk factors are not well understood at all for things like testicular cancer. It's one of those things that just happens. There's that. What I read, the little bit of reading I did on it, said there were no understood risk factors. But it is one of these ones that, if it's caught early, and it typically is because of the pain involved, then it's one of the more curable cancers. Well, moving towards the outside here, we're going to talk about the epididymis. The epididymis is this little coiled structure which lies sort of on the margin of the testes. It doesn't actually have anything to do with production of sperm. It stores sperm for a short period of time. There's some indication that it may actually, in some way, manage to pick out, sperm which are not quite formed right or something like that, replacing, removing the damaged sperm, and also adjusting to a slight extent the composition of what's referred to as tubular fluid. We'll get more about that in just a second. So it's just a temporary storage apparatus, and it's real obvious on our slides. It's like, on this one, it's this coil. These are the seminiferous tubules in here, and this would be the epididymis right along the side here, or in this one it would be like right there. So anyway, it's real obvious. Temporary storage spot, and then moving towards the outside, we go into the ductus deferens or the vas deferens. This is the tube that carries sperm to the outside. So we start in the testes, then the epididymis, now the ductus deferens. 16, 18 inches long is usually the length given for it. It runs initially as part of the spermatic cord and then sort of breaks off from the spermatic cord and runs sort of by itself. And I'll show you the picture in just a second. It joins after, let's say, 16 inches, it joins with another structure called the seminal vesicle to form a very, very short little structure called the ejaculatory duct, and then we'll get to what happens after that, the prostate gland and all that kind of stuff. It has a little pseudostratified columnar lining with a little cilia on the end of it. We're not going to pay a whole lot of attention to that. But a surgical procedure called a vasectomy, which I'm sure you've all heard of, is a procedure for male sterilization. So if you, well, let's use this chart. So here's one ductus and here's the other ductus. If those are just snipped or ligated, cut, whatever you want, then no sperm gets to the outside, can't fertilize the egg. So you're not going to be having any babies, right? So it's a quick and painless kind of method for birth control, which under pretty reproducible conditions, however, can be reversed. So sometimes a man will have a vasectomy and then whatever, something changes in his life or whatever, and it's going like, you know what, I kind of maybe like to have maybe a baby or something like that, whatever. And using little microsurgical procedures, the tubes can be kind of sewn back together. Because here's the weird part about it, and some people don't know about this. I actually had a math